iRODS impact kind of writ large on science and data management?
Thanks, Daryl. I wish I were smart enough to talk about iRODS impact writ large. That's what we thought we would do, but of course I found out that I can't do that. So I'm really going to talk about two very specific projects where where we're using iRODS pretty much, I think, almost out of the box with maybe a few additions that we've made. So we'll see. The beautiful part of it is that the people who actually did the work, or the one person who did most of the work, Mike Conway is here, so hopefully if you have any really good questions, we can turn to Mike. Okay, so we want to, like I said, I'm going to look at two use cases, Brainy, and I think that Mike had mentioned Brainy before, and the other one is a system called SC2I. So I'll show you how we are using iRODS in both of these cases and go forward. So Brainy is also a neuroimaging data. You heard another talk earlier about neuroimaging data. There's an initiative that was launched. I think it was not just in the U.S. but also in Europe, called the Brain Initiative, which is to sort of try and get a much better understanding of the structure, functioning of the brain, and how it impacts disease, cognitive abilities, etc. And part of that project has led to a number of different imaging methodologies, because both for the structure of the brain as well as the function of the brain, which is believed to be essential by neuroscientists to be able to understand functioning of the brain. So I'm not a neuroscientist, but we're working with one. And so I'll tell you a little bit about that. The second one, SC2I, is related to being able to deal with data from caring for wounds. And as we get into it, understand a little bit more where it's coming from. Okay, so I'll return back to this image. It's kind of a low resolution version of actually a mouse brain, if you will. Okay, so in neuroscience, there's the technologies that are being used to image are such that now it is possible to produce very, very large data sets at very high resolution of the images of the brain. The ultimate goal in neuroscience is to create this connectome, which essentially shows every neuron in the brain and its synaptic connections to all of the neurons. So you actually have a map of how everything gets connected together. Okay. The way that a lot of the brain imaging used to be done to get the three-dimensional structure was actually to take very fine slices of the brain, and then you image each of the individual slices, and then you have the problem of how do I now stitch all of these together and to create a three-dimensional image, and that's still going on. But what has also happened recently, and this is the particular technology that we are working with, is that there are new brain clearing techniques which have come into existence. In particular, the one we are working with is called Clarity. And what that allows you to do is to actually go through a chemical process which takes a couple of weeks, which basically removes all the fatty tissue from the intact brain and replaces it with a transparent material. So you don't no longer have to take sections of the brain, but rather you have the brain structurally frozen in a medium through which you can actually pass optical light. The problem with trying to do that otherwise is that the fatty tissue just reflects light and you really can't get the resolution you need. So this was a way of taking out the fatty tissue and replacing it with this other material. And then what you do is you actually have an optical sectioning process. So you don't actually physically section it, you put it in the microscope and you go through and you create an optical section. So for a mouse brain, which is not that large, each of these images can result in, each of these scans can result in a data set that's about 4 terabytes just for that one image. A typical study that these folks undertake may involve 20 or 20 mice which have some sort of knockout, genetic knockout, and then 20 wild type mice, and they want to compare them. So all of a sudden, even a single study, you're looking at several hundred terabytes of image data. So that's an example of what you have on the left-hand side, the Chung Nature 2013 article. And that was probably, I think that article was the first time that this brain clearing and imaging technique was described. So some of the data problems you have with these types of images, and there's other imaging modalities, and I'm not going to talk about that. But some of the problems you have is that you want to be able to share and move this data, all the typical things we have been hearing, searching for data across labs, compute. So what you want to do with these images is to actually go in and find each body of each of the neurons, as well as perhaps some of its dendritic connections, trace them. So the image processing to be able to do that is fairly computationally expensive. The most successful methods are based around deep learning, like everything else now in the world seems to be based around deep learning. So those are some of the issues that we need to address here. Okay. So the BRAINY, as we call it, the BRAINY project, which stands for Brain Infrastructure, is currently funded by the National Science Foundation. And what you want to do is to be able to, it's a creation of a computational infrastructure for storage, sharing, and analysis of such 3D images, in particular, the clarity cleared light sheet images. Then be able to actually trace the neuronal structures using novel image processing algorithms, and then finally to be able to visualize this in 3 dimensions. So that's the sequence of what you want to accomplish. So what we are doing as part of the BRAINY initiative is to try and create a more comprehensive cyber infrastructure, and I'll show you the different components. No laser. Okay, so on the left-hand side, you can see that there's an instrument computer where the data is being acquired, and you want to be able to move. Typically, that moves into a laboratory computer, and then you want to ingest it into the BRAINY system itself. Then you have some analytics application which need to go into some sort of maybe a remote compute server, maybe in the same facility. Some visualization applications. And then there's a variety of other functions that we are in the process of creating, don't yet have. One is to be able to do some search for these images, search through these brain images based on some suitable indexing. So there's a data discovery and sharing that we are working on. Another one is to have data citation and persistence. In many of these cases, the general model that the scientists want to follow is that once the images are acquired, they want to embargo the images for a certain period of time while their lab works on the images to create the publications needed. But more and more funding agencies, such as the National Institutes of Health, for example, say that you can have an embargo period, but after the embargo period, the data has to be made available widely to other researchers. So, once it's made available, how do you make it discoverable? And also, if there are results that you are publishing, how do you make them persistent so they can be found afterwards? How are you able to cite them using some sort of data object identifier? There are a number of labs around the United States that are able to do these imaging methodologies. So you really want to be able to federate across the different data archives that are there. For example, you might even want to do it like the Allen Brain Atlas has got a large collection of brain images, and you might want to be able to link with some of those. be able to back up and replicate the data. And finally, this notion of data scoring, which is based on, you'd like to be able to produce some properties of the data which gets reflected in the data discovery. Some of the data scoring properties could be based on, how frequently was the data set accessed, et cetera. What's the size of the data, maybe some metadata associated with the instruments, et cetera. Those would all be sort of data scoring, but equally what we are trying to build here is a social component where other researchers can come and access the data and be able to score the data, maybe give it 1 to 5 stars, and that also gets reflected in the search mechanism. So this is the overall structure that we aren't completely built yet, portions of it have done. Okay, so the data ingestion process, Mike Conway is the one who has been working on it, and so there's basically an agent that lives on the instrument computer and pushes the data out into a laboratory iRODS grid from which it is moved into the brainy at a later point in time. So here is an overview of that. And we are in the process of ingesting some data right now. Is that correct? Okay. So these data sets they take a while to actually create, to be actually imaged, to go through the clearing process and image, and so we are getting our first sets of images into Brainy as we speak. Okay. One of the interesting things that we are also doing during the data ingestion process is to be able to make the process of attaching metadata to that image as simple as possible through the use of metadata templates. The idea here is that if you, like the experiment I described where you have maybe 40 mice that you're going to image for a study, there's a large number of metadata-oriented parameters about the study which are common across all of these specimens. And there may be a few that are actually particular for each individual mouse brain. And also there may be instrument settings that are the same and also may be different across the different images that you created. So the idea here is that you want to get as much data as you can from the microscope itself and fill it into your metadata template, as well as you have the researcher fill in other things into the metadata template, and then you try to capture that, and they only have to fill in specific information for a few types. Okay. So that's the metadata ingestion process. As you can see, here is an example where the breed, for example, the genotype and things like that are all going to be common, and then a few other things are specific to this. So try to automate the metadata capture as much as possible. Okay. And then eventually, of course, all of this goes and lives in the iRODS data grid that exists right now. Right now, this data grid exists at UNC Chapel Hill, if you will. Okay. In terms of analytics, I think I am running out of time, so I'm going to go fast. We have made use of the Cyverse discovery environment, so you can actually have an application that is packaged as a Docker container. You have the discovery environment web-based interface. You can go in there, select the data, select the app you want to use or the analysis code you want to use, and fire it off to do the analysis. Right now, we have some code that we have created that can either run on an HPC system or an HPC with GPU clusters. Here are some examples of the different times it takes to analyze one mouse brain, if you will. And then the big advantage for the scientists themselves is that the process of moving the data to the GPU or the HPC system and back is completely transparent. They don't see it at all. The researchers also want to be able to do some interactive visualization of the images. So we have set up a way that they can actually use things like ImageJ, which is a commonly used tool to be able to look at the data. And this is done using WebDAV. Is that correct? Yeah. We have plans to provide access using Jupyter notebooks and be able to maybe create some notebook-based reproducibility. So I'm not going to show you this. There's actually a little visualization here so we are basically using Oculus to be able to look at the details of the mouse brain, and this is a little video of that. I'm not going to show that, but just to tell you, the right side is the front of the brain, the mouse, and the back, and the left side is the rear, and that's sort of the density of a certain kind of inhibitory neurons, if you will. We are actually replicating this at more than one place. So the kill devil, what you see on the right side, is actually closer to where the laboratory instrument itself is. So the data first is going to get ingested in there, and then get replicated over at another facility. Okay, so that's the Brainy. Tell you very quickly about the Surgical Critical Care Initiative. It's called SC2I. So this is a project in which it's actually funded by the U.S. Department of Defense, by a bunch of military hospitals. And what they're most interested in is that when to be able to do wound care based on the physiological and genetic makeup of the person who has been injured. So it's a clinical decision support system where somebody is wounded and comes in, and by looking at their phenotype and genotype, they can decide exactly what is the right care that should be provided to them. So there's a bunch of military hospitals that are providing this data, and there's also a bunch of civilian emergency rooms that are providing this data. The data is, it's a large variety of data. It needs to live in the cloud. So the U.S. government has something called the GovCloud, which is really a zone in Amazon Web Services that has special security provisioning. And so there's a central data repository that holds all of the data, and we are developing the central data repository for this project. So data from all of these places gets deposited into a landing area in GovCloud, and then iRODS rules are used to ingest it into the CDR and also to do data cleanup as it happens. Justin here is the one who has been working on this, Justin James. After the data is in there for analytics, there's data extraction that takes place, and again, iRODS rules are used to control who has access to the analytic data that's pulled out and used to do various studies. So I'm going to stop here. Okay.
So Dan's going to come down. We'll load him up. All right, coming to you. Can you pass this back? I'll throw it.
Throw that far.
Hi. Thank you for the presentation. The question that I have is that you show that you have this agent running on the microscope PCs.
Yeah.
Are users at that point already able to determine whether the data they've collected is actually suitable for ingestion?
Right now, I believe that the data just gets ingested, so the suitability of the data has not been determined at that point, and the idea is that the reason why we have this ImageJ interface is that that's what the researchers used to go in and play around with the data and see if it is suitable or not. So the agent just takes it from the microscope and pushes it into the grid, independent of whether at that point the data is suitable or not. Is that right? Is that? Yeah.
But are you then not afraid of ending up with lots of test data, lots of not suitable data?
It well could be that we may have to change our policy on how exactly it gets ingested. The data is just starting to come in, so we really don't have a lot of experience as to how many bad data sets just get ingested and then thrown away very soon, and why not save ourselves the trouble. From what I believe that, the first look that they take at the data while in the instrument computer is very cursory, and the expectation is that, the researcher uses the instrument, the microscope as a resource, and the people running the microscope want the data off of that as soon as possible. So the amount of checking you can do while it's still on the instrument is limited, and you would have to do it after you move it somewhere. Okay. Make sense? Anything else? Anybody have questions?